Okay, in this video I'm going to explain the process of protein synthesis, which is the production of proteins that takes place in the ribosomes. And in order to understand this, you first must realize that the recipe to build this protein actually comes from the DNA that's in the nucleus. And if you recall, the DNA, which has the recipe, is not capable of leaving the nucleus because the pores that make up the nuclear envelope are too tiny for DNA to pass through. So the DNA needs a little help to get the message that it contains to the ribosomes. So what I did here is I drew a picture of an unraveled piece of DNA. And in my unraveled piece here, I went ahead and wrote down a very specific combination of bases on the left side here. Now, in order to get the DNA out of the nucleus, like I said, the DNA is much too large to fit through the little holes called the nuclear pores. So we must find a way to get this message out of the nucleus and to the ribosomes. So what I'm going to do is I'm going to utilize a molecule that is called mRNA. And the M stands for messenger. So in the nucleus, where my little drawing is taking place right now, this left-hand side is the side that has been identified as holding the recipe for the protein that I need. So in order to copy this recipe, I'm going to utilize nucleotides that are RNA-based, meaning that instead of using T's, these bases are going to have U's, and when they come in, they're going to pair up A to U, C to G. So this message would be transcribed as A, U, G, U, U, A, G, U, G, G, C, U, U, A, G. This process that I just did by copying down messenger RNA to this strand of DNA is what we call transcription. Remember, transcription takes place inside of the nucleus. After this has been copied, or shall I say transcribed, the next thing that happens is the messenger RNA is going to detach from the DNA and make a journey to the ribosomes. I'm going to go ahead, and this is my big giant ribosome that I have drawn right here. So what I'm going to do is I'm going to copy this message that I've written right here, as if I pulled this out like a little train. I'm going to copy this down. So what I get is I get AUG, UUA, GUG, GCU, UAG. If you'll notice, I put these in sections of 3, and I did that for a very special reason. What we call these sets of 3 bases we call these codons. Now, in order to know what a codon means, I have to use a chart, and that's what this chart is right here. The way we read this is we find the first letter in the messenger RNA to the left, the second letter at the top, and the third letter to the right. It's the combination of those 3 letters, or codons, that give us a specific amino acid. So what I'm going to do is I'm going to take the combination of codons that I just put into my ribosome down here, and I'm going to go ahead and I'm going to translate the message. So this process that I'm doing right now that takes place in the ribosome is called translation. Now, in order for translation to take place, I have to utilize another kind of RNA. This RNA is what is called tRNA, and the T stands for transfer. Now what's going to happen is the transfer RNA molecule is going to read the codons, and when it sees the codon that agrees with it or matches up with it, it's going to go fetch the amino acid that it needs. So what does a transfer RNA molecule look like? Well, on the transfer RNA molecule, there is what is called an anticodon. The anticodon is the opposite of what the codon is. So if this were AUG, the opposite of AUG would be UAC. So this transfer RNA molecule, when it sees this AUG, it's going to go off and fetch whatever AUG is on the chart. If I look on my chart right here, if I take the A with the U with the G, what I get is a start codon. Now this start codon actually has a name as an amino acid. It's called methionine. And you see this right here. And what this will do, this transfer RNA molecule is going to go off, and it's going to go fetch a methionine molecule, and it's going to come down and drop it off right here. My next codon is UUA. So if I look on my chart, for UUA, there's a U, there's my second U, here's an A. I get an amino acid called leucine. And so I'm going to go ahead and draw, there's my transfer RNA. The anticodon would be AU, and attached to that is leucine, and that's going to be brought down here. And when it gets dropped off, it's going to form a bond with this other amino acid. This bond is what is known as a peptide bond. So this right here, I'm going to go ahead and write that down for you. This is a peptide bond. My next codon is GCG. If I look at my chart, there's a G, there's a C, there's a G. That's alanine. And so alanine is going to be fetched by a transfer RNA molecule, bonded by a peptide bond. And then if you look again, there's my GCG. Again, that was alanine. My next one that I need is GCU. So if I come onto my chart, GCU happens to be another alanine, and it's going to bond with a peptide bond. And my last one, UAG, if I go to my chart, there's a U, there's an A, and there's a G. I get a stop codon. The stop codon is what tells, and again over here was a start codon, the stop is what tells the ribosome that the recipe is over, it is done, and that is all that is needed to produce this protein. So this isn't a very big protein at all. In fact, this is completely fictitious, but this would be a protein. As you can see, I've got amino acids bonded together, and once they get bonded together, they will twist and turn into the shape that happens to be for that particular protein.